Hello, dear students. I am Ashwin Saxena, Assistant Professor, School of Pharmacokinetics, IFT University. Today, we will discuss about the new topic, Basic Pharmacokinetics and Compartment Modeling. In Basic Pharmacokinetics and Compartment Modeling, we will discuss firstly pharmacokinetics. Pharmacokinetics is defined as the kinetics of drug absorption, distribution, metabolism, and excretion, and their relationship with the pharmacological, therapeutic, or toxicological response in man and animals. There are two aspects of pharmacokinetic models. First is therapeutic aspects, which involves development of pharmacokinetic models to predict drug disposition after its administration. Statistical methods are commonly applied to interpret data and assess various parameters. Second is experimental aspects, which involves development of biological sampling techniques, analytical methods for measurement of drug concentration in biological samples, and excretion and evolution. Here is a plot between plasma drug concentration versus time profile. It is a typical plasma concentration time profile showing pharmacokinetic and pharmacodynamic parameters obtaining after oral administration of a single dose of a drug, in which when a plot between plasma drug concentration versus time profile, they showed the two parameters. First is pharmacokinetic and second is pharmacodynamic parameters. In pharmacokinetic parameters, 3 points may be considered. First is plasma peak plasma concentration Cmax. Second is time of peak concentration Tmax, and area under the curve AUC. The peak plasma concentration Cmax generally depends upon administered dose, rate of absorption, and rate of elimination. The second parameter is pharmacodynamic parameters, in which they generally evaluates minimum effective concentration, maximum safe concentration, onset of action, onset time, duration of action, intensity of action, therapeutic range, and therapeutic index. In pharmacokinetic modeling, drug movement within the body is a complex process. The major objective is therefore to develop a generalized and simple approach to describe, analyze, and interpret the data obtained during in vivo drug disposition studies. The two major approaches in the quantitative study of various kinetic processes of drug disposition in the body are model approach and model independent approach, also called as non-compartmental analysis. It is a flowchart which shows the different pharmacokinetic models is divided into compartment models and non-compartment models. Compartment models is further divided into physiological models or mechanistical models. Physiological models which applied on whole body, partial body, or metabolism. Then second is pharmacokinetic model approach. A model is a hypothesis that implies mathematical terms to concisely describe quantitative relationship. Pharmacokinetic models provide concise means of expressing mathematically or quantitatively the time course of drugs throughout the body and compute meaningful pharmacokinetic parameters. The general applications of pharmacokinetic models in the field of characterizing the behavior of drugs in patients, calculating the optimum dosage regimen for individual patients, evaluating the risk of toxicity with certain dosage regimens, correlating plasma drug concentration with pharmacological response, evaluating the bioequivalence between different formulations of the same drug, and explaining drug interactions. Types of pharmacokinetic models. Generally, there are of two different types, compartment models and second is physiological models. Compartment models are also called as empirical models and physiological models are very realistic models. Compartment models, compartmental analysis is the traditional and most commonly used approach to pharmacokinetic characterization of a drug. These models simply interpolate the experimental data and allow an empirical formula to estimate the drug concentration with time. Depending upon whether the compartments are arranged parallel or in a series, compartment models are divided into two categories. First is Memorelli model, second is catenary model. The Memorelli model is the most common compartment model used in pharmacokinetics. This is a figure in which different types of Memorelli models are explained. First is model 1, in which one compartment open model intravenous administration. When the tourist form was given intravenous route, the absorption phase is bypassed, goes into the system circulation and then excrete from the different pathways of the body. Second is one compartment open model extravascular administration, which is generally applied for oral and rectal route dosage forms, in which the absorption phase firstly started, then goes into the system circulation, and then excrete from the different organs of the body. Third model is two compartment open model intravenous administration, same as one compartment absorption phase is bypassed, then goes into the system circulation and parallelly and reversibly goes into different organs of the body. Fourth model is two compartment open model extravascular administration. Fifth model is three compartment open model intravenous administration. And model 6 is three compartment open model extravascular administration. Memory model is the number of rate constant which will appear in a particular compartment model is given by capital R. In the case of intravenous administration, the value of R equal to 2n minus 1. In the case of extravascular administration, the value of R equal to 2n, where n equal to number of compartments. Second part of compartment modeling is catenary model. A figure which shows the catenary model is a series of compartments which are joined to each other. When the dosage form goes into the systemic circulation, then first phase is absorption, then goes into the different compartments of the body, and then one by one goes into the series of compartments, and then excretion is followed. And the second model of pharmacokinetic modeling is physiological model. These models are also known as physiologically based pharmacokinetic models. They are drawn on the basis of known anatomic and physiological data and thus present a more realistic picture of drug disposition in various organs and tissues. The number of compartments to be included in the model depends upon the disposition characteristics of the drug. For physiological models in which the dosage form when goes into the system circulation, they are parallelly goes into the different compartments or organs in the body through the blood, and then finally excrete from the body. Thank you, all of you, for cooperation and silence.